All right. Well, I'm very happy to be here. Thank you, the organizers, for having me here. So let me talk a little bit about, I guess, what was it? The title that they gave me was Simple to Complex Models and Mechanisms of Arrhythmia, Historical Perspective. So I tried to come out with a talk that fits the title they gave me, so that's good. A lot of the stuff that I'm going to show is in collaboration with my wife, Elizabeth Cherry. She's actually teaching right now, so she'll be here at 4 in the afternoon. So, but a lot of the work here is done in collaboration with her. So I think it's almost like we did it together. This is kind of weird. This feels funny. I guess what we're trying to do this workshop is trying to understand what produces a spiral wave, what spiral wave breaks into multiple, what are the mechanisms, and then how can we control them. So, so in order to do that, I guess we have mathematical models, and we can go from simpler ones. Actually, just to mention this is a pig heart. This is also in optical mapping, so it's a voltage spiral wave produced, and then after a few seconds develops into VF. So for simple models, one of the simplest models that I can think of limit cycle model is actually is order 0 of what we have for an ion channel that opens and closes non-linearly, allowing the flux of ions. It's actually considered two containers. I brought a couple of things that I don't think I'll have time to do it during the talk, but after the talk I can show you. So one of these things is just a container like this. So you have two containers, one inside another one. They're fixed, and they're only connected by a small hole. So you have a hole that connects the two containers. If you fill this one with salt water, the inner container with salt water, the other container with fresh water, and you put them at the same height, then what do you expect to happen? The inner container has more density than the higher density than the lower, so it's going to leak. So it's going to leak into the freshwater container. So it's going to be, if the hole is small, it can produce a laminar flow that goes down. And if it's laminar flow, there's not too much mixture, so it comes down and it saturates in the bottom, and it just stays there without mixing. So the level of the water starts to rise. Once it starts rising too high, then the pressure changes. Now you have more pressure outside than inside, so now you have a flux of fresh water into the salt water. And again, it's a laminar flow that then saturates in the top. So as long as the gradient remains, there's going to be actually an oscillation of this flux. So this is actually kind of like if it was an ion channel, but it's driven by a Taylor instability. But still, it's non-linear and allows the flux of periodic oscillation, that the periodic oscillation can change as function of the parameters of the system. The same way you have a parameters that you can change in a ionic channel, you can change the thickness, the radius, the thickness, the ratio between the two containers, the density, and all those parameters can change the period of the oscillation. But that's just the very first part. This is work done actually in one of my students, Diana Chen. You can change the parameters to get actually a shape of, you can actually, this because they're salt, you can put an electrode inside in each container and measure a potential. So you can actually see a voltage potential between the two. And as the oscillation goes, you can have an action potential. And as you change the parameters, you can change the action potential to make shape to look similar to an action potential. So in here, one is the Selene oscillator and the other one is a sinoatrial node. Can anybody guess which is which? Which think this is the Selene oscillator? All right. So okay. So this is the SA node, and this is the Selene oscillator. So this is. So you can actually, one of the things you can do is then you can put a syringe pump that you can program, so you can add and subtract water. So that is the perturbation in the system. So our balance subtracting is the same as perturbation that you can excite the system. So it becomes from a excitable system from oscillatory system to an excitable system, and you can perturb it so you can pace and pace at faster frequencies. And as you pace, there's actually similar as in cardiac tissue. There's a period doubling bifurcation. You go from a 1-to-1, which is a constant APD, to a 2-to-2. We have alternance between the rhythm of the oscillation. Let me show you also, you have multi-stability. You can have, you have the oscillations, but at for some parameters, you can have actually both fluxes at the same time. So that actually behaves as a fixed point. So you can have an oscillation like in cardiac tissue. Some cardiac tissue, some SA node cells, you can actually perturb it, change the phase, and you can make it actually stable. So similarly with this system, so it's all very similar. It has the same dynamics as a cardiac cell. You have oscillation, you can perturb it, and you can get a fixed point. And from the fixed point, you can also perturb it and go back to the oscillatory system. So you go between the fixed point to the limit cycle and vice versa. And as I say, to stop. It's not stopping the oscillation. You go into the both instead of oscillating the rhythm going up and down, then you can have both fluxes at the same time. So it's leaking both at the same time like that. So in that case, the voltage that you measure is 0. So that gives this limit cycle. This fixed point, and then no, by the time of the perturbation. So same as in the cardiac experiments, if you perturb at the right phase with the right stimulus, you can kill the oscillation. So similarly, you do the same thing here. So it's oh, that's good. Thank you. So this is, and then as I say here, you can, as in cardiac tissue, you can perturb fast, and you can get alternates. So it's a very nice system. It's just basically a zero order of a cardiac cell. And so since we're talking about the simplest models possible, this is one of those that you can use. So you can do a mathematical analysis of this, and you can do also an experimental analysis of this in the lab. Yeah, so actually it's very nice. You can actually, all these dynamics, you can get it by using a Fitzo-Nagumo, but instead of order 3, order 5th. So by order an order 5th, then you can have two basins of attraction, and one that is limit cycle and another was a fixed point. Well, it's just some phenomenological version. You can have a phenomenological version that is just a Fitzo-Nagumo. Then you can do a more physiological model that accounts for the salinity. Yeah, it's a fast and a slow variable. So in this case, it's just a fast and slow variable that gives you, the fast variable will be the voltage.
So it's not a physical?
No, it just will be a phenomenological version, yeah. But you can do a phenomenological model out of NIMBioS talks. So for NIMBioS talks, you can get a more phenomenological model. Good, actually. Yeah, if you have questions, just stop me. So let me show you. So anyway, so same thing. You have a simpler, and actually I have the oscilloscope, so we can do it after the class. We can show it and we can see it. Same way you can have a simpler model for the cell, you can have a simpler model for spiral waves. This is one of the simplest ways you can do is just collect a bunch of students. You can get 600 students in Georgia Tech, and you can put the premature stimulus. You can just initiate with a pulse that is broken, and then you can just initiate a spiral wave. So it's a very simple system to initiate a spiral wave. So you can see the more students you have, the larger the wavelength you'll have, but at least 600 is enough to have one. Yeah, we put it on YouTube, yeah. Only this one, but not the next one. The next one, one thing that happened, even though Georgia Tech students are very smart, they not always follow the rules exactly. So it's hard to see sometimes yourself, I mean, the signal coming from behind. There's a little bit of decoupling. There's also students get a little excited when they see the wave coming. So then they get excited before the wave should come. So then it's a little bit of small wave coupling and a little bit decoupling by the side. So that's enough, actually, just a small little changes like that. You can produce actually a spiral breakup. So you have the spiral wave here, and at some point you'll see a couple of little activations at some point, and the spiral wave breaks, and it goes into chaos. So this is a very simple example that shows just coupling. Very small changes in the coupling can actually stabilize a spiral wave and can result into complex chaos. So again, so those are the two simplest cases I can imagine for starting from there, going up into complexity. Any questions? Not yet. It was very hard to actually do, but so this is part of the Atlanta Science Festival. We did it. It was last year, first time. So now that get people excited, we'll try it next year again with more, and then do more complicated stuff. I want to do rules that include restitution, so you can have this effect. So we did a lot of different examples with pacing from the corners. Also, there's a certain amount of time you can keep the students' attention. So we did a few, and then we tried to do the simulations to reproduce experiments from this. But yeah, so the idea is now to do more complicated dynamics to them. Um, anyway, so the use of simpler models, actually, the first simpler model of cardiac dynamics was actually produced by Wiener and Rosenbluth in 1946. Wiener was a mathematician who was also happened to be very handsome. He was a very handsome mathematician. Yeah, when I saw that picture the first time, I was like, wow. He actually did this work in Mexico, so I had to ask my mother. It's like, did anything happen? No, it turns out I only have the looks. I don't have the brains. But and this is Arturo Rosenbluth. So both work in Mexico and develop a method. It was actually probably the first mathematical computational method of studying biology. And actually they started, so they're considered the fathers of cybernetics. From this paper and another paper they did in avionics. One of the things that Arturo Rosenbluth used to say, he was so he was a mathematician, he was a cardiologist, so he's one of those big so in a meeting like this, it's good to collaborate between really the two different regions of science. And so he used to say that when analyzing systems, the best model for a cat is another cat, preferably the same cat. So it shows actually the variability in biology, right? And we have to do actually how models intrinsically are probably not correct by design. So you have to do approximations to see whatever you're trying to study. The next modeling, that was the first model actually of a PDE, an ODE, and then connected to as a PDE in tissue, of a cardiac cell was by Dennis Noble in '62. He did a Purkinje model. And then also in '62, around that time, also Gordon Moore was using cellular automata producing interactions of spiral waves and breakup due to basically heterogeneities like the one we saw with the students. A lot of the dynamics actually at the time also in the '70s, it came from Puccino in Russia. And actually this is one of those cases when they say everything has been done before in Mother Russia. This is actually true. They did a lot of stuff in Mother Russia. Let me just show you a little, I'll show you just a few minutes of these movies. It's a really amazing movie. Actually here when you start seeing, so back then, they actually tried to simulate the Fitsunagumo in the 1970s with these kind of machines. And then to visualize, whenever we had trouble to actually visualize our data, imagine what they did. They actually just did ASCII printouts. And they start doing activations.
Young people, that's called green bar paper.
Green bar paper. So without screens, nothing, they did simulations. Back then of Fitsunagumo in 2D, and not only the waves, they then analyzed that actually you can have initiate spiral waves by heterogeneity. And they did analysis on hearts. So I can show you later on the movie if you like. The whole thing is about 15 minutes movie. It's very interesting about how they basically they did a lot of the studies in Puccino when Valentin Krisky was the director. He pushed a lot about this, the analysis of this and experiments. He got the reason they started doing this is because of the Belousov-Sabotinsky reaction. So originally it was Belousov. Sabotinsky was in that lab there. And the main contribution that Krinsky did is that he asked him to do the experiments that he was doing, reproduce what Belousov was doing, but then also do it in space and do these patterns in space. So the spiral waves, they saw them in the chemical reaction, and then that's how they went into doing the experiments in simulations, and then come back to the experiments in small hearts. And actually, dog hearts too. They put electrodes, and then they could actually later on put the pictures of the spiral waves on top of the electrodes that they measure, and they were very so they really discovered all these spiral waves back then in 1970s. Anyway, so this is just and of course Valentin and Sabotinsky, they got the medal of Lenin medal for that. Yeah, one was there, but he went there later, and then he took it. He brought it. So Howard Winfrey is the one who brought it back to this side of the continent. Anyway, so We can go with simpler models like Fitsunagumo or a 4-variable model or 3-variable model. We use, with Alan, we developed a 3-variable model that it was fast enough at the time. The computers were not that fast, but we were able with 3 variables run simulations in 3D and study that the effect of the fiber rotation can actually destabilize spiral waves and produce elongation on the vortex filament and produce breakup. So that's one of the examples of producing simple models. Simple models, actually 4 variables is enough to reproduce a lot of the dynamics of without memory of restitution and shapes of action potentials of more experimental data. So this experimental data, this is the model underneath. It can fit to other complicated models like the 10 Toshers. So this is a 4-variable model fitted to a model that is 16 variables. No, this was the second one, so 24. So you can reproduce actually the same dynamics that you can observe between the frames. Another examples of things you can do with simpler models is you can study dynamics and compare like in tissue very quickly. This is an example of 2 models, 2 simplified models that they have the same restitution curve. They only have different shapes. So one is more triangular and the other one is a little more square. But when you measure the APD at a constant constant threshold, they have the same behavior. So if you do the simulation in single cell, you don't distinguish between the 2 models. The 2 models have the same restitution, and because there's no memory, they follow the slope of the restitution that they produce alternance. But when you put it in space, the electrotonic effects. When you put it in space, you have electrotonic currents. The electrotonic currents, the diffusion becomes actually a current that affects the dynamics of the cells. And I'll show another example later on. So in this case, the 2 models in single cell, they have the same dynamics, but in space, one is stable and the other one produces breakup, just by pure coupling. So one enhances alternance and the other one suppresses alternance and leaves a stable spiral wave. And again, we're going to see through the whole workshop that alternance is a mechanism that produces breakup. So when you pace fast, you go from a 1-to-1 to a 2-to-2, and eventually goes into VT and VF. Another example here, another simpler example to study is, for example, the zebrafish heart. It's very small, it's easy to access, and the action potentials are very close to human, actually, in duration. Even though it's a small little heart, you can get about 300 milliseconds of duration. And like the novel model, this is actually the novel model of 62. When you pace it at fast cycle lengths, there's a period only bifurcation. It goes to 2-to-2. And then actually this model, because the slope of the restitution changes slope later on, you can have also a 1-to-1 later, so you have 2 regions of alternance, separated by a region of conduction block. So this actually happens for the zebrafish. The zebrafish, you pace fast, you get a very beautiful period bifurcation. You get a long action potential and a short action potential. And like in the models, really long, really short, really long, really short. And this is the experimental data that we have. So it grows like that. It grows actually as a square root, as a pitchfork bifurcation. So it grows out very large. I mean, you can see the alternance magnitude is really huge, right? It goes from 140 to about 50 milliseconds, right? So it's a huge change in alternance. Now, but if you compare to a mammalian heart, this is a dog heart. These are microelectrode recordings that we did in the lab. These microelectrode recordings on dog hearts. This one, when you do it on the heart, in the dog heart, the amplitude is much smaller. The amplitude that you get is about 10 milliseconds. And not only that, when you have alternance in these models or many models, mathematical models, you have long and a short. The short one is just basically an abbreviation of the large one. But in mammalian tissue, it's not actually the case. It's not that it's smaller. There's actually a crossing. The small one is actually larger at a higher potential, and the larger is actually smaller at the higher potential. So they cross. They cross, and the amplitude is small. So this is some of the differences difference in species, difference due to the dynamics of the calcium and the voltage. So those are some of the differences that we have to account when we do simulations. When we create models, models have to account for the different correlation between voltage and calcium. Yes, even between, yeah. So I'll show at the end difference between atria and ventricle. Yeah, so there's a gamma, and also you have difference between epi and endo. Alternance often occurs faster on the on the endocardium and later on the epicardium. So even for the same cycle length, the point of bifurcation occurs faster first in the endo than on the epi. So that's a big difference. Another example that shows the effect of physiology actually is when you have, this is also canine restitution, this is dog data, this is a restitution curve when you have normal tissue. So you get a little bit of slope greater than 1 and you get alternance. But when you take out the calcium, when you do an experiment without calcium, if you use thyroid and you use calcium, so hypokalemia, then actually the slope gets greater than 1. The slope actually can get up to almost 3, but there's no alternance. So it gets much steeper than before, but the alternance disappears because it's produced by calcium. And in that case, it's effect of memory, and you can measure it very nicely. So this is the when you have memory, instead of getting the slope greater than 1 as the effect for restitution for alternance, it's actually a combination of the slopes between the S1 and S2. So this is something Elena did a long time ago. Very nice work. So this is her derivation of when you have slope greater than 1. So when you do the experiments in the dog with low calcium, you actually can reproduce these curves and calculate the slope. And the slope of the S1 is the steady state is steep. But when you account for the effect, then the relationship is below 1, and it gives you stable. So this explains why, actually, in this case, when you have low calcium, the mechanism of alternance is not produced, even though it gets much steeper restitution. Another thing for account is the heterogeneity of the hearts and the tissue. So we have Purkinje fibers there for dogs, for example, in mammalian hearts. Well, different mammalian, even within mammalian hearts, there's difference. So small species like dogs and cats, you can see that the fibers, when we stain them with Lugos, you can see them very well, and they're like branches. They start and they branch out. I'll show you another picture after that. But and the dynamics you can get one of the Purkinjes you can stimulate from the side, and you can measure with microelectrodes, and you can see the alternance. Again, it's not like in some of the models, like the novel model that slopes grows as a pitchfork bifurcation. It grows more linearly like a pitchfork, which is something that also Sean Penn has been working a lot over the years. You can have a simplified model that reproduces the dynamics. One of the important things is that even though if you have two models that reproduce the data, so this is the data of the restitution, you can fit the data with a continuous curve, or what some of the theory that Sean Penn has, that is maybe fitted with an effect of a border collision. So a border collision is given when you have it's continuous curve, but the derivative is discontinuous. So it has a little kink. So if you do the fitting of this data with any of the two curves, when you see them, you see both of them give you the same fitting. It's a good fit. But if you do the iteration of these two curves and you get the alternance, you get two different effects. You get that the point of bifurcation at which starts is different, and also the amplitudes can be different. And you can compare with the experiment. So the experiment, if you do the border collision bifurcation, just this effect here, just one little change in your fitting, that matches the data exactly. You don't do that, you completely miss the whole dynamics of the alternance. So this is an example that even a simpler model, you have to have the right mechanisms underneath to reproduce. If you don't have the right mechanism, well, people may say it's because it's too simple, but it's not only because it's too simple. You have to write the right mechanism to reproduce the dynamics. So for example, even more complicated models, the Francesco model later on, that's a second generation model, accounts for a lot of the intracellular models, intracellular calcium. So this model actually, once you pace fast, it doesn't produce alternance at all. It loses the effect of alternance, and actually, even though it gets really unphysiological results. When you start pacing too fast, it goes really, the resting membrane potential goes really high, and the amplitude also decreases. The Asaldi Enol model of 2009 is 30 variables, and still also it doesn't produce alternance. And the Lee and Rudy model of Purkinje model, also 38 variables, and it produces alternance, but very little, and only in single cell. When you put it in a space, then it doesn't produce alternance anymore. So more complicated don't necessarily means better, at least in this case. It doesn't reproduce the dynamics you observe of alternance. We have also changes in scale. You can go, actually, I was going to show this. Let me move this one here out of. So this actually scales. We can go a lot of different scales when you analyze the heart. This heart here is a neonatal heart. This is of a mouse. This is a mouse heart, full developed mouse. And then this is a horse heart. So you can see the difference in scales between the modeling. And then you can have, of course, all the range in between, right? So this is dogs, this is sheep, and these are two are horses' hearts. You can have. Now you can see the difference also in Purkinje. This Purkinje network here is from horse. So it's very different than, so ungulates have a different distribution of Purkinje than, for example, dogs or cats. The distribution here is actually a network. You can see that the crossing, and it's really, really complicated compared to the. Yes, but it was more like it starts from so it starts from a bundle, and then the bundles just spread out. And then there's a little bit, some places around here, just really, there may be a little crossings, but there's not as much crossing as when you see here in the, here, let me show you a couple of pictures. This is another horse heart. So you can see it's very complicated, the network. They actually connect, disconnect. So that makes a big different level between the two systems. So modeling becomes very complicated, right? It's not only the physiology, but also the structure, the length scales, and the physiology of the structures. It's hard to know. It's hard to know because then you have to do it with a microelectron, and then know exactly when you go from one to another. But when you do it, even when you do it in bundles, when you do it in bundles like that, and you go. Sometimes one microelectrode you see as an action potential, you go deep next layer, you can see actually a completely different action potential. So they're actually decoupled sometimes between the nerves. So when they cross, they may just cross on top of each other, so may they cross and then actually may be connection. But that's something we don't know, and that also needs to be considered for modeling. Actually, doing studies and doing optical mapping on these systems is really hard because it's hard to stain the Purkinjes. The Purkinjes, because they are above, they're not easy to stain. And if you want to stain them really well, you have to put more dye, and then it becomes toxic, and it's really, really hard. So I only know, I think just Guy Salama is the only one who has done a couple of Purkinje examples and with optical mapping, and it was not. It's probably also to activate better. I mean, to go. I mean, horse heart, you have to go really. You have to spread activation much quicker everywhere. So the crossing may help. But again, those are some of the questions that need to be responded by simulations, right? And
so maybe another variable there is how far into the tissue the muscle junctions.
Right. They go, right? So the horse and the ungulates go deep into the tissue, and humans and dogs, they stay in the surface. So that's another big effect and for example, there are experiments when you can do optical mapping experiments when you measure the unit and arrhythmia, then you can kill the Purkinjes by using Lugos. You can kill them, and then the arrhythmia changes, so it becomes a little less complicated when you take out the Purkinje network. So when we go back to modeling cardiac cells, again, you can start with simpler models like 4 variables. You can go to like Rudy, 8 variables. You can go to the Ayer model, 67 variables, much larger. Actually, when they published this one, they published a table with initial values for different cycle lengths, because it takes so long. They took them so long to do the simulations that they put actually values of the initial conditions for different cycle lengths, so people will know it. But then again, a lot of people who develop models like this, they're not experts in simulation, so they only do in single cell. They do in MATLAB, so it takes forever. You can do better integration algorithms, and you can do them in space, in tissue, and run them much faster with a better still result, but much higher delta T's. and you can do simulations better. And then that's when you realize that actually a lot of these models, even though they're complicated, they reproduce a lot of the data they were used to reproduce the experiment. So they reproduce a lot of IV curves and the restitution. But when you go into more dynamics in space, then they fail. So let me show just you a few examples of that. So human ventricular models, there've been an explosion of models over the last few years. So right now, there's the previous vocal model was the first one, 1998. It's 17 variables. Then 10-Tosher came out in 2004 with 17 variables. Then there's another option, a second version of 10-Tosher 3 years later that has 24 variables, a little better calcium dynamics. The Ayer model, it came in 2004, 67 variables. There are 65 were Markov models. Everything was Markov model except one gate variable and the voltage. So only 2 ODEs, the rest Markov models. This is a phenomenological model I did with a student of mine and with Elizabeth. There's the Grandi et al. Model that came in 2010, 38 variables. And then the latest also is the O'Hara, 2011, 41 variables. So these are the models that exist so far for human ventricles. And then if we analyze them, they actually all have very different dynamics. This is the action potentials for them in single cell, and then when you put them in space. So also, as I was telling you, once you put it in space, you have the electrotonic current. And that electrotonic current, you don't often think about it. You always think as an extra current, as extra value that is there. But if you think about it, it's a current. It's really a current that is there. And as a current can affect the other currents non-linearly. So it really can change the shape of the action potential. And you can see a lot of those actually, even the latest, the O'Hara-Rudy model, which is the latest, it completely, once you put it in tissue, completely loses the notch. There's no notch. There's just completely goes up and down. So once you put it in tissue, then who knows what you're actually modeling. You can see also the difference in durations, right? It goes all over the place. Not only shapes, but durations. This one here is an action potential from microelectrode recording in human. Of course, the 4-variable model we did that fitted to that one, so that's why it fits that. So it's biased, but still, it just you can reproduce that.
Is there a critical size, and you need to set the size of tissue or fiber
to show that? No, actually, it turns out that very small tissues can actually, once you couple, it's just very few. Probably like 5 or 6, and depending on the discretization. But I'll show you an example that it was very mind-puzzling, and it was the first time we realized how important the electrotonic effects are. But yeah, just only a few cells is enough to have that coupling, right? And of course, as I was saying, depending on the shape of the action potential, is stronger or less this current, right? If you have a triangular action potential and atrial, it will be not so strong. But if you have more a ventricular, which comes down faster, that one has a much stronger effect, right? The Laplacian gets much stronger. Yeah, but still the space constant is not actually, when you measure it with the simulation, still it depends on your discretization. It depends on the space constant that you can measure from the experiments and the one you see on the simulations is actually a little different. But yeah, I agree that it has to go related to how many cells you need to do that.
If you have a short fiber, then the APD is almost uniform.
Mm-hmm.
Short fiber.
Right.
But I don't know about the shape of that.
Yeah, but they will have effect, and then also once you have a small fiber, you have boundary conditions, and also that may change affect or more or less the dynamics. Yeah. So here actually show for all the models, we show the different currents and how much is the strength for each current. So even though they're all supposed to be for the same type of cell, which is human, you can see that the distribution of currents is completely different between the models, right? Some of these one, for example, this one, this is ITO. No, this is IKR. Sorry, IKR is pretty dominant for this model versus for this model here, and anyway, so the distribution of currents is very all over the place. These are the APD restitutions for the different models. So they're all over the place. I mean, really, really, not only on magnitude, but also in shapes, right? So even if you were to rescale them all, still will be the shapes are very different. Some are very flat, some are more steep. So the dynamics is all over the place between all the models. Same for the conduction velocity. Actually, the worst thing is the O'Hara-Rudy, which is the latest model. It has a very, very slow conduction velocity. It has a very unphysiological conduction velocity. So the sodium they had, it was really not correct. It gives you about 38 centimeters per second conduction velocity. And you expect to have something around 70 along the fiber, along the fastest axis. This is S1, S2 restitution curves. Again, so this is different restitution curves for different models. So some have more memory than others. This model has very little memory. This model has a little bit more memory. Flat restitutions versus a little bit more steep. So we can quantify them more, but it's just showing you a little differences along them. As you put them in tissue too, you see the difference, right? This is a epicardial model in 0D and in 1D. For the endocardial, you can see a big difference. Actually, you see a difference between the resting membrane potential between the two. This model produces alternance for a long region of cycle lengths, but once you put it in space, the alternance disappears, right? So it's not the same, the dynamics in space than in single cell.
What's the difference in the model between epicardial and endocardial?
They change some of the currents a little bit. So the conductances of some of the currents change a little bit. Some parameters, yeah. For example, ITO is more dominated in epicardium than endocardium, so they can have the notch. So they tune a little bit some of the conductances to get. The equations are the same, yes. It's just only parameters a little bit different. This is when you simulate it in tissue. These are the simulations of the. This is the Ayer model. This is the Peeble-Bockelman, the 10-Tusher, and the 4-variable model that we have. So you can see that this is stable spiral wave, this is quasi-stable, and this is breakup. And this one actually, that's supposed to be the more accurate because it has 67 variables to reproduce all the currents. It breaks over the place, but not only breaks a lot, the frequencies at which it breaks, it goes very unphysiological. It has frequencies of, at the same time, of mouse and human through the same simulation. So you see the action potential gets really, really tiny. So basically, once it turns the spiral wave and turns, the calcium gets inactivated. There's almost no calcium, so the action potential is really, really, the wavelength is very small. So it gets completely unphysiological with the calcium. These are simulations for the other two models, the Gandhi and the O'Hara. So those ones actually also give a quasi breakup. But then again, so there's no consistency between the models. Different models give you different dynamics. Unexpected. This is a summary of some of the differences and some of the shortcomings of the models. They don't match experiments. They don't match the AP amplitude. The maximum restitution slope values. The obstruct velocity in tissue is not correct. The restitution of the slopes also is very variable between the models. Same thing happened for example, canine ventricular models. We can compare the two that are the Fox et al. Model and the Hunt-Rudy. The Fox has 13 variables. The Hunt-Rudy has 30 variables. We can do a similar analysis. The shape, actually, here, this is the main difference. Once they publish, so when Rudy published this model, then later on we did the simulations, and they had to publish this model later on because when we simulated in tissue, this is the Hunt-Rudy. The original Hunt-Rudy is the in single cell is the solid line. When we put it in tissue, then suddenly we got this action potential. So the first thing we saw is like, oh, we make a mistake in the code, right? So we went back and checking where's the error, but it's only the Laplacian. So we were there like for a day. And then eventually Elizabeth, she just started decreasing the size, the coupling and the size. And eventually she saw close to the pacing site, you were recovering this original shape. As you start moving away, just a few cells away from the pacing site, then you get this shape. And what happened is that in that model, the sodium again was very low. So in single cell, it was enough to produce an activation. But once you put it in a space, the sodium was not strong enough, so it didn't go as high. So you can see that the original one is the dash side, and the solid one is the one in tissue. So once you put it in tissue, the sodium didn't go as high. So that slowed down the L-type calcium channel. It didn't got activated in time, so it got delayed, and that's why it enlarged the action potential. So it's just a few change on the sodium, delay the L-type calcium, so elongate the action potential, and elongate it almost by almost 100 milliseconds. So completely different dynamics. So when we did that, so then we had to send an email to Rudy and to Joram, and we spent like a day writing, make sure that it was not offensive and making it. So he got it, and then he decided to change the sodium, and that's when he got the next version of his model. And again, so the difference between the two models on the different concentrations of the magnitudes of the currents, they're completely different between the models, so they are not similar. Again, the dynamics of alternance is different between the models and different between single cell and tissue. So again, this is single cell. This is 1D for the three models. This is the Fox model. It produces alternance actually that terminates, but here when you put it in tissue, actually it grows a little larger. In the 0D, the 0D model, when you do it in 0D, you get alternance. When you put it in tissue, it disappears. This newer version is a little bit more similar in space. Anyway, and again, this is the difference in alternance that I was comparing with experiments too that I was showing. This is when you get alternance in the two models, the difference between the two bits is just a reduction. It's just only like scaling in time. But in real experiments, when you do it in optical mapping or in microelectrodes in dogs, then you see the crossing. Actually, it's not that the long and the short is just only a scaling in time. It's actually a whole shape dictated by the difference in ion channels at the time. This is the simulations of the two spiral waves. And sorry, I don't play this one. But both of them, they give spiral waves, but different lengths of the tip trajectories. Anyway, so this is some of the summaries of these of the differences. Anyway, and there are also the ATR models, the human ATR models. There are like 3 human ATR models. We have made the comparisons, but I don't want to just show you again. It's just same effects. They are different. The dynamics between the 2 of them, the 3 of them are qualitatively different. Same for rabbit ventricular models. Same for Purkinje models. So all these models, we have characterized them, and then they can always have differences. So, and then they fail sometimes to reproduce experimental data. So that's where a lot of work needs to be done to quantify these models, to see where they fail, and hopefully try to see how we can make them change to reproduce experiments. So there are basically 2 main problems for modeling when we, in addition to the ones you talk, is basically that I would like to mention. One is the need to speed, and the other one is reproducibility of results, right? So speed, once you have all these models with very complicated equations, you have 30, 40, 60, 67 for Ayer. The latest version of the Samson Purkinje model, I think it's about 90-something equations for one cell. Once you put them in space, then it becomes very computational intensive. So this is work on 2 of my postdocs, Greg and Ezio. He finishes now in Austria in a position. So we're doing now simulations of these models, but then using a GPU. So with GPU, actually, you can run, as long as the models are not too complicated, small models you can run almost close to real time. They run really, really fast. So there are different options to do GPU. So the hardest thing is programming in CUDA. So Robert Gilmore, I used to work with him. He used to say that it was very funny. He said, before you spend all your money on supercomputers, now you have to spend all the money on the programmer to do the programs. So, yeah, the computers are cheaper, but now you have to spend all because the coding is much harder. But now actually there's a new option to do. So you can do this with CUDA programming, but also there's OpenACC. There are libraries that if you have a program that you have it either in C or in Fortran. We do a lot of Fortran actually. You can just do these libraries. It's very easy. It's like almost like MP. But this is just for graphic cards. So you can put it in, and then you can convert your serial call into parallel. And it doesn't get the speed of CUDA, but it gets the speed at a big speed. So still with the difference between CUDA and OpenACC that we've been doing different models, this is still an order of magnitude, but it's still the difference between single processor and OpenACC is a couple of orders of magnitude. So you can see the difference, and it's very quickly to do. So I encourage you to, if you go to do simulations in tissue, to use graphic cards. Graphic cards are in many devices, even cell phones. So then you can run actually your simulations in the cell phone. So you can run, this is in my cell phone, you can run and you can interact. You can produce spiral waves, conduction blocks. So it runs, you can run, I'll show you if you want at the end of the sim. So you can run, this is a Fitsunagumo 512 by 512. So 2 variables. So this is actually half a million differential equations every time step that the cell phone is doing. And it runs real time. Well, it runs, like you can see the spiral will rotate. You can do 3D simulations too. In the cell phone, so you don't actually need a supercomputer. You can do it in the cell phone, but and actually you can even run the 10-toucher in a cell phone. So you can run it. It takes 24 variables, 94 equations. It's about 6 million ODEs every time step. So it takes a little longer to rotate, but it takes probably like a couple of minutes to do a rotation, but still you can do it in the cell phone, so that's amazing. So if you can do that in the cell phone, imagine you can do with a good graphic cards, right? So now because you can do really fast simulations with a fast graphic card, you can do actually sensitivity analysis before, like people are doing it in single cell, but now you can do it actually sensitivity analysis in tissue. So you can do very quickly. This is not only 512 by 512 because sometimes spiral waves don't fit. So this is 1024 by 1024 cells with a discretization of 250 microns, DT of 0.01, 50 rotations to make sure that that's stable, right, for each one of the elements. And that takes about 3 hours to do one of these panels. So that's really really nice nowadays. This is work with Amir Najid. So just to terminate, to finish a little bit more talking about some of the work that still needs to be done is to get the models to reproduce some experimental data. You reproduce a lot of experimental data, but there are some key experimental data that we still fail to reproduce. So I'll show you quickly some alternates and some fibrillation. Let me just move this one out. So this is, again, experimental data from a Purkinje fiber. And this is experimental data optical mapping on a dog ventricle. So you can get discordant alternance in this small cable of 2 centimeters. You can get discordant alternance in about 2 centimeters. And the conduction velocity in a Purkinje is about 300 centimeters per second. It's very fast. The conduction in tissue in dog is about 80 centimeters per second. And you can have like 3 nodes in 3 to 4 centimeters. So small domains can handle actually alternance or discordant alternance. And actually, I was going to show you this is not typical example, but it still is possible. So this is still also a dog ventricle. For some cases, you can actually have a lot of nodes, even in 3 centimeters, you can have a lot of nodes of discordant alternance. So the length scale actually is very small. It requires very small. But if you try to do simulations in these models, they actually fail. They don't reproduce. If you want to do discordant alternance in a model with 300 centimeters per second velocity, you require, actually the minimum tissue required is about 12, 13 centimeters compared to 2 centimeters here. So that's a big failure of the models. The same for the ventricles. The ventricles is a little lower the velocity, but still you can fit discordant alternance maybe like in 7 centimeters, but you can fit in 3 centimeters, right? So there's a discrepancy of what the model can do and the experimental data. So that's basically the coupling. So there are a lot of tried solutions to do that. The coupling, there are some studies that have done maybe a fractional Laplacian. So assuming that the coupling is not really homogeneous. So there's discretization effects. So then the coupling is by a fractional diffusion. That allows a little bit decoupling. Another study we're trying to do is with Claire. This is here. She's trying to do an analysis of assuming that from experiments, the gap junction is a function of the transmembrane potential. So if you assume that, you can actually have, like in experiments, that as the action potential forms, you can have decoupling during the plateau. So if you do that in a model, you can keep the conduction velocity and APD restitution the same in the model because it doesn't affect during the upstroke, but during the plateau, the conduction changes. So if the conduction changes during the plateau, it decouples, so that allows more freedom to get these discordant alternates. So this is another mechanism that can probably be included into models to reproduce more experimental data.
Is there experimental data
Yeah, this is experimental. Yeah, but now, but also this is the voltage function of the change in. So you have this change, but also you have a time dependence. How quickly it goes to this shape. This is steady state. So the model that we work in, it reproduces this, but also the time constant that we're using is not necessarily what we've actually seen experimentally. So it may not be exactly what it is, but it's one mechanism to try to fix the problem. And then maybe it's a lot of it's effect of this plus many others, plus maybe this if you combine this with the fractional diffusion, with hepatic conduction, things like that. All these thing effects together may allow for this to account then to get these discordant alternance in the models to fit as in experiments. Huh? Monodomain. Yeah, yeah. That's another thing. I forgot to mention that I was going to put a slide. Another effect is the bidomain. The conductance of bidomain. The people use only basically there are 3 values that people use in the literature, and I forgot I was going to put a table there. They vary completely between the 3 different values that people use. They vary dramatically. So which one to use is a problem. So we need to actually get, and we're working with a mathematician in Emory University to try to use experimental data to calculate all these conducting effects. So we need to get for the bidomain, even in the monodomain, the right conductances for the diffusion. So that's another effect that needs to be done. Oh, because it's very hard to get here the value, because it's V over I. Yeah, this is experiment from actually from in Rochester in Syracuse. What's his name? It's a physiologist in Rochester in Syracuse. No, not Arcady. Vance something. I forgot. Should have put the reference here. Vistra. Yeah. Thank you. Yeah. Thank you. Anyway, so yeah. So this is just an effect when they're trying to calculate it. It just goes because you divide by 0.
That's actually what you expect, right? Because when you think about, you're basically talking about the flux that's driven by some gradient, right? Gradient doesn't have to be voltage. Gradient could be caused by a whole bunch of other things. You basically pose a gradient in any concentration to get a current. So what you get is actually what you expect, right? You should expect. Basically what you measure here, you think about resistance.
Yeah.
Basically the resistance. For an electric current, there is no other source for current other than the potential difference. For two cells, that's actually not the case. Even if there is no potential difference, you will still get a current if you vary the concentrations of the different cells.
It depends on whether the ions is going in the cell or go out the voltage. Is that, I don't know.
So
You keep the concentration different, but the same potential. You'll get a current.
So they're calculating here is the current over the voltage to calculate this curve here, and then they have 2 cells connected, and then they fix one, and then they vary the other one. But then when they get to 0, when they get voltage 0, then they have they're divided by 0, so that's what the problem is.
That's fine. Is on this gap, right? So I can understand that I can do some experiment on this side, on the negative voltage, some of the positive voltage, and expect them to meet each other, right? Because I don't understand why there's a gap.
Yeah, we can talk, but it should match. I mean, theoretically it should match, but it's just because experimentally you're dividing by 0 the voltage, so your values start going higher and higher. Numerically you can get a limit, but experimentally it's harder to do that. All right. Anyway, another mechanism is of alternance or no alternance. So now Ilya, one of my postdocs in the lab, he finally set up optical mapping for voltage and calcium at the same time. And it's it's a complicated issue because you have to put the light synchronized with the cameras. You have to use the right lenses, the right actually, it's a lot of, we find in the literature, there's a lot of mistakes of how people are been doing this. You really want to have zero crosstalk between voltage and calcium. So for that, you have to get the right filters at the right frequencies of stimulation. So you get actually, for when you stimulate, for the calcium, you stimulate at the value where the voltage doesn't change at all the signal. So you have to avoid any crosstalk. So it's a lot of background theory. Took more than a year to get it running, but now it's very good. We get good signals. So this is a rabbit heart. So you can get voltage signal is for this dye changes negatively in relation. So in this case, voltage is the signal that goes down, and calcium is the signal that goes up. So this is spacing a rabbit heart, spacing at slow frequencies. If you go to fast frequencies, then you can see very nicely, you can see the alternates. So you see long short on voltage and long short on calcium, right? But that's what we see experimentally, and that's what you have seen in a lot of papers, and we always see, and we're trying to model. But now we start getting access to more other different animals, and you can see the differences here. This one, this is a pig atria. And here you can have alternates in the calcium, but almost no alternates in the voltage. Right? The voltage is almost no change, but there's a good change in the calcium. Now that's, you say, okay, it's very little, right? But here, yes, so that's what I'm saying. So you have to put two dyes, one dye for voltage and one dye for calcium. And then they both get excited at different frequencies, but they emit at the same frequency. You take a video, right? So you take a video, and then you can see. So the spiral wave I showed at the very beginning of the spiral wave in the heart, that was optical mapping of the voltage, just using this. But now we can, at the same time, we have voltage and calcium. So we can measure with one frame, every even frames we can measure voltage, and every odd frames we can measure calcium from the same point. So there's no crosstalk, there's no two cameras. Some people sometimes have used two cameras, but it's hard to then to synchronize the two regions. So it's the same camera, same point, zero crosstalk, and then voltage and calcium. So in this case now, you can see from that region, you can see, so the alternates here. Let me show you then that was the alternates that you see that I was showing you. Some alternates in calcium, a little bit, almost nothing in voltage, but you say, okay, it's very little calcium alternates. But this is in pig atria, this is cat atria. Here you can see really there's no difference on the voltage, and then you can see the difference in the calcium, right? This is really long, short, long, short. So now this is a mechanism that is not even in the simulations, we have not even something like that, right? So now we're starting to get data that produce that. You can have also the other thing, differences between atria, right? This is pig atria, and this is pig ventricle. Pig ventricle, actually, it's what we actually we should strive to be. We should strive to be have a pig heart. If you pace really fast, you can pace up to 160 milliseconds, and it's 1-to-1. It doesn't produce alternates. So there's no alternates in voltage, and there's no alternates in calcium. So this is this is all different mechanisms that you can try to study now numerically, and with the models try to get mechanisms that reproduce the dynamics. The other example that we fail to reproduce still is when we do simulations of fibrillation. Every time we do fibrillation, you take any paper. So this is just two examples of paper. This is from this is Krex and Rickers, and this is from Ed Wigmont. Experiment simulations in fibrillation. You always see, no matter what model you use, you always see that you get, of course, complicated dynamics on the action potentials, but you always go back to rest state. In the models, you always go to back rest states, right? You never go to the higher. When you do experiments, and again, this is another. This is actually not to say the other models. This is actually my simulation. So again, just to say we fail. I mean, we all fail, not just. Still goes back to zero. But if you take, of course, optical mapping is a case, but if you do all your microelectrode recordings, still the signal goes above and the signal goes below. So it never goes back to the rest state. And that's something no model actually reproduces yet, and needs to be done. So the experiment.
Well, I mean, maybe the delay constant in the instrument is not that enough.
No, that's why, for optical mapping, I agree. But if you do it microelectrode recordings, you get the same thing. And microelectrode recordings is, you can run at 10 hertz. You can get signal from microelectrodes at 10 hertz. It's not that you're skipping bits or skipping data. It's that. So there are two effects. One is that the signal doesn't go back to rest state, and the other one is that the sodium doesn't go as high. So both of them are decreased. Yeah, 3D. Yeah. So even when you do 3D, and with fiber rotation, you may get closer a little bit. Some of these one closer, but you still get beats every now and then that go down to the lowest. So anyway, so those are some of the effects that I think there needs to be done, and I'm perfectly out of time. So then just want to thank, of course, Elizabeth Cherry, which more than half of what I presented is hers. So thank you, Elizabeth. She'll be in this afternoon. So Ilya, Greg, and Ezio, three of my postdocs. Stefan Luther, I've done a lot of low energy defibrillation, so I didn't have time to talk about that today, but I'm happy to talk later on with anybody. Robert, which is the one who we can blame to make me an experimentalist. So I really thank him a lot, Robert. Niels, which is here. John Bragad and Laura Munoz, which is here too, collaborators. My students, especially Claire, which is here. She's done a lot of the voltage, the gap junction. Diana, she did, and Amir, and Jairo. This is the lab, and I'm asking for questions. Thank you.